and welcome back to Nutrition for Cancer Prevention, Lesson 3, as part of the MUIH Professional and Continuing Education course. I'm your presenter, Jason Bosley-Smith. Thanks for joining us for this third and final part in the Nutrition for Cancer Prevention course. This evening we'll be looking at and exploring the evidence basis for chemoprevention of various individual nutrients and herbs. Our learning objective for this content is to examine the molecular and biological functions of individual nutrients and herbs in the context of the hallmarks of cancer that we've discussed in previous lessons 1 and 2. So we'll explore each of these individual nutrients, herbs, botanicals, and compounds, nutrients, and discuss their broad physiological and biological activities, their biochemical activities, and specific pathways by which they may inhibit carcinogenesis or tumorigenesis, as well as provide some protective effects against the damaging compounds and the damaging effects of that can lead eventually to development of a cancer process, namely things like an inflammatory cascade or damage to cell and metabolic processes, cell structures, and various other structures of what would be normally a healthy cell that would shift it into, again, what we've discussed in lessons 1 primarily, a rogue cancer cell, a cell that's going into some form of dysregulated activity, mitochondrial dysfunction, and eventually bioenergetic fluctuations that, essentially set things up for a cancer cell or a tumorigenesis process to begin. We'll begin this evening by discussing sulforaphane. And sulforaphane, many of those in attendance may be familiar with this compound. Highly regarded, highly well studied, the precursor glucoraphanin. Glucoraphanin, as the precursor, converts to the isothiocyanate sulforaphane via the enzyme myrosinase. Myrosinase is actually found in the plant structure itself. It's found in our GI tracts, and basically is developed via a variety of gut bacteria that can actually, based on GI kind of status and GI health, is one of the things that will activate and convert over the precursor of glucoraphanin to the active form of sulforaphane. So with the myrosinase being inherent in the plant, it's actually somewhat unstable when high temperatures are affected. So once temperatures via cooking can actually deactivate the myrosinase enzyme, making conversion of glucoraphanin over to sulforaphane a bit more challenging and not as efficient and not as robust. So an important component is to potentially address GI health in a patient to ensure that they can create that conversion, and also consider the temperature by which cooking would occur, or combining both cooked sources of glucoraphanin with those that may be raw as well. So we'll talk about some sources. Clearly the ones that most everyone knows are in things like broccoli and broccoli sprouts, but all cruciferous vegetables, mustard greens, turnip greens, kale, cauliflower, they all contain some of these precursors in glucoraphanin that eventually can give us this wonderful compound of sulforaphane. Sulforaphane is, as I mentioned, very well studied and has shown a variety of anti-cancer properties and activity. It potently inhibits the neoplastic cellular processes, which just fundamentally would, of course, lead to carcinogenesis. And one of the main primary activities it has is by upregulating the phase 2 response and antioxidant response elements. So phase 2 response enzymes, primarily that pathway is cytoprotective. So when we have a compound like sulforaphane that can upregulate or induce the phase 2 enzymes, been proven to be a very powerful and highly efficient strategy for protecting against damaging effects of ROS or reactive oxygen species and intermediates, many of which, again, we've talked about in prior lessons, can preclude some form of pathogenesis, some form of dysregulation of cell function that could lead to cancer. There's a variety of phase 2 enzymes that sulforaphane can upregulate, and by doing this, it really kind of helps to, with the gene expression, upregulating this gene expression. So this compound, as well as several others, have been shown to regulate transcription, upregulate enzyme activity, and provide a protective effect against the harmful effects and damage to cells. So as you can see in this study, the chemopreventive activity of sulforaphane, published in Drug Design, Development, and Therapy, this has a variety of clinical studies it's discussing here that have supported the chemopreventive effects of sulforaphane on carcinogenesis. There have been both clinical phase 1 and phase 2 studies the authors are referring to that have showed that broccoli sprout extracts containing sulforaphane were well tolerated and caused no significant adverse effects in healthy volunteers, women with breast cancer and men with recurrent prostate cancer in these sort of cumulative studies. In addition, there have been some recent studies, this particular paper is discussing, where the reports have shown that sulforaphane effectively inhibited tumor growth and increased sensitivity of cancer cells to chemotherapeutics in patients with advanced pancreatic ductal adenocarcinoma. So not only having some protective effects, chemopreventive effects, but also being able to work synergistically based on some of the literature around working with chemotherapeutic drugs that were being used in specific cancer populations. So the authors sort of summarized by saying that based on the above mentioned studies, it is clear that sulforaphane is a safe and relatively non-toxic chemopreventive agent and exerts anti-cancer activities through multiple mechanisms, including regulation of phase 1 and phase 2 DMEs, or drug metabolizing enzymes, anti-inflammatory activities, influencing cell cycle arrest, induction of apoptosis, important to prevent, again, damaged cells from continuing to survive when going through a possible conversion to a cancer cell, and the epigenetic regulation of several different types of kinases, cyclin-dependent kinase, and several of the other protein kinases mentioned previously. Okay, so the next nutrients or individual compounds here, the herbs that we're going to talk about, two constituents here that both fall in the group of polyphenols. And these polyphenolic compounds are, again, probably very popular and well-known amongst the clinicians listening this evening, as well as even with patients. Many patients are familiar with the general evidence and the general use or benefits around curcumin and EGCG. So we'll discuss each of those independently. And we'll start with curcumin. And as, again, many of the course may be familiar with, the curcumin is that active compound ingredient derived from turmeric, curcuma longa, the turmeric root, having a variety of well-established literature around anti-cancer or chemopreventive types of properties. And again, there's the primary curcuminoid, curcumin, as well as a couple of other curcuminoids that seem to have some specific effects around the pathways and hallmarks of cancer that we've discussed in prior courses or prior lessons. So dimethoxycurcumin and bis-methoxycurcumin, both two additional curcuminoids along with curcumin itself that appear to have some of these biological activities. And based on this particular study, looking at some meta-analyses and cumulative studies here, curcumin, an active component of turmeric, curcuma longa, and its effects on cancer, the anti-cancer effects. Here, the researchers, again, to highlight that curcumin has been found to be effective in many phases of cancer development to suppress transformation, beginning development and invasion of tumor, angiogenesis, that development of new blood vasculature, and metastases. Curcumin has been determined to suppress the growth of tumor cells via cell proliferation pathways, cell survival pathways, capsase activation pathways, tumor suppressor pathway, death receptor pathways, and many other cell signal pathways that contain protein kinase pathways as well. And protein kinases, just to sort of reinforce here, are the enzymes that regulate a variety of biological activities of proteins by phosphorylation of specific amino acids, adding a phosphate group typically derived from ATP, that energy molecule, which induces a conformational change in the protein structure into a different form. Protein phosphorylation itself is known to play an important role in a variety of chronic disease processes. It's involved in various cell processes, cell division, metabolism, survival, apoptosis, And as I mentioned, protein kinase has been found to be involved in pathological processes and specifically in malignancy. So you can see here curcumin having a direct effect on some of these signaling pathways that are often associated with some form of or some proliferation effect of the cell structures towards cancer. And sort of noted here in this paragraph by the researchers was that the curcumin has been shown to have impart its effect when decreasing or preventing various cancer types, and they list several. So this is not just indicative of one particular cancer type, but here they list multiple myeloma, cancers of the colon, pancreas, breast, prostate, and lungs. So pretty extensive effects and pretty essentially long-standing literature, quite significant in the research around the effects of curcumin and those different curcuminoids as chemopreventive agents. Next, we'll move on to EGCG, or epigallocatechin gallate, and this is, again, an active constituent we normally associate with green tea. And here we're looking at multiple pathways by which EGCG can impart its anti-cancer activities. There's certainly a good bit of epidemiological evidence around the consumption of green tea and lower incidence of cancer, but mechanistically here there's a mini review published in Molecules and Cells, which talks about some specific pathways by which EGCG imparts some of those anti-cancer properties. So here in the cancer prevention with green tea and its constituent, EGCG, some investigations from human cancer studies, and if you see on the right-hand side of your screen, a variety of different sort of graphically represented pathways where EGCG is protective of the cell, blocking some of the effects of tumor promoters, of endothelial growth factor, with the bottom arrows indicating the sort of outlying outcomes here, inhibition of metastasis, having a synergistic anti-cancer effect when combined. So often using, again, some studies going back to what we just explored with curcumin, some synergistic effect when used as an adjunct to standard care of treatment with chemotherapy. So all leading to sort of cancer prevention globally. And again, this is just one example of a pretty robust mini review that considers several studies, but there are many of these types of reviews and meta-analyses that show the same effects of EGCG in chemoprevention. And here you can see on the next slide, we talked early on in lesson 1 about the idea of cancer prevention from someone who is not undergoing cancer process, but also considering secondary and tertiary cancer prevention, so prevention of recurrence of cancer. So here, this chart really kind of speaking more to that green tea for the general population may have some influence, and this is from the researchers discussing in that prior study, looking at the potential for delay of cancer onset based on its biological activity, and then for both general population and cancer patients, secondary cancer prevention, so medical activity, early diagnosis, early treatment. So if there was then a cancer process that occurred, if there was a cancer diagnosis, for cancer patients following cancer treatment, again here showing green tea coming back into the fold, green tea itself or green tea plus anti-cancer drugs, again looking at sort of an adjuvant type of relationship here, providing some evidence of prevention of recurrence and prevention of second primary tumor and metastasis. So working on multiple pathways, both for primary prevention as well as secondary, tertiary prevention. Moving on next to another series of polyphenols that appeared to have some chemopreventive properties, quercetin and apigenin. So again, we'll go through each individually, look at some of the research and literature, and look at, again, some sources where these could be derived from a dietary perspective, and maybe where there's some evidence for potential therapeutic dosing, or at least, again, from a fundamental preventative or prophylactic standpoint, incorporating these compounds into a healthy overall diet for cancer prevention. Quercetin, again, probably a relatively popular compound and polyphenol. It has antioxidant capacity, is allergic or antihistamine properties, antioxidant, antiviral, a variety of different things where quercetin has really sort of a robust effect across a number of different biochemical activities or pathways. And here we're looking at a review of the anti-cancer potential of quercetin, a comprehensive review looking at a number of these. And the researchers are citing, again, some dietary sources of the flavonoid. We've got everything from high amounts in onions, apples, also finding quercetin in cilantro, dill, some other herbs, berries is another great source. And really here, again, cumulatively looking at this review, researchers looking at the effects of the anti-cancer or chemopreventive effects of quercetin, discussing here in the paper that quercetin impedes the propagation of various types of cancers such as lung, prostate, liver, breast, colon, and cervical. These anti-cancer properties are exerted through various mechanisms that involve cellular signaling, so going back to some of the other mechanistic properties we saw in some of the earlier compounds, and the ability to inhibit enzymes responsible for the activation of carcinogens. So again, some of the ones we mentioned, protein kinases and other enzymes that are going to influence the potential for carcinogenesis. Quercetin, they go on to say quercetin displays anti-cancer effects based on its binding to cellular receptors and proteins. And the researchers conclude by discussing that in addition, quercetin has been reported to have synergistic effects when combined with chemotherapeutic agents such as cisplatin, which may further improve the outcomes of a traditional chemotherapy. So going back again, similarly to what we saw with curcumin to EGCG, that not only does quercetin have some direct biological effects related to chemoprevention in and of itself, but that it actually has the ability to work synergistically with traditional conventional treatments, in this case chemotherapeutic agents specifically, to provide a more synergistic, robust, and extensive effect of chemoprevention. Moving on to apigenin, another maybe lesser known than the first few that we've discussed so far in this lesson, but maybe a slightly less known polyphenol, but a compound that is found in a number of different types of foods, and again has pretty extensive evidence for its anti-cancer properties. So apigenin is another polyphenol. It's found in a number of sources of food. We'll show a chart in a minute. It's certainly found in some of the most high sources in parsley, for example, and various other foods and some other herbs. But parsley is sort of the king of apigenin. It has a relatively high concentration of apigenin relative to some other food sources. So in this review, again, looking at apigenin in cancer therapy, the anti-cancer effects and mechanisms of action, researchers cite that apigenin has been demonstrated to show broad anti-cancer effects in various types of cancers, including colorectal, breast, liver, lung, melanoma, prostate, and osteosarcoma, and that this flavone inhibits cancer cell proliferation by triggering cell apoptosis. So as we've discussed early in lesson 1, triggering the appropriate programmed cell death, which again is one of those hallmarks of cancer, evasion of apoptosis by a cancer cell. So addressing that here with apigenin, inducing autophagy. So again, that idea of essentially cleaning or recycling cellular debris and cell structures that can be damaging to the cell. So inducing that, and also modulation of the cell cycle, which of course dysregulation of the cell cycle is one of those sort of features that we see in cancer as well. So apigenin affecting cell apoptosis, autophagy, and the cell cycle. So having a relatively broad effect there on the potential for cancer. Also discusses here in the paper that apigenin decreases cancer cell motility and inhibits cancer cell migration and invasion. So particularly interesting here and useful for the concept of not just a localized tumor, but the potential for metastatic disease. So directly inhibiting that cancer cell's ability to sort of extricate itself from a local tissue and to metastasize to other areas of the body. So again, to further kind of support the additional benefits, the researchers mentioned that apigenin was reported to show anti-cancer activities by stimulating an immune response, so also inducing some immunomodulatory effects and enhanced immune activity through apigenin. So multiple pathways here, pretty interesting to see that apigenin has such a a wide array of effects on a variety of processes that are all really important to protect or inhibit cancer development, and if there is cancer, as I mentioned, also prohibiting that cancer from migrating to other areas of the body. So during those processes, the researchers discussed that multiple signaling pathways and protein kinases are modulated by apigenin, and it lists several. So that's the same idea. We're seeing some similar mechanisms of action here as we did with some of the other nutrients and polyphenols that we've already discussed. And here's a nice graphic. Again, this is from that same paper we just discussed some of the finer points on. This is a nice figure here in figure 2. You see apigenin, and with the arrows kind of pointing down, they're indications as bold arrows of apigenin inducing a particular outcome or pathway. And with the longer sort of T bars signaling there or representing the suppression of those effects. So if we stick to the top 3 arrows with the sort of light red or pink boxes, we can see that apigenin induces apoptosis, autophagy, and immune response, like we just mentioned, and a number of different pathways and enzymes indicated under each of those. And then also going down to the boxes that are in the blue, that is suppressing the effects around cell cycle, which would be dysregulated, and suppressing migration and invasion. So again, really impressive to see something like apigenin as this polyphenol compound being able to affect multiple pathways associated with cancer. So transitioning into this chart, you can see here that a variety of foods which contain various amounts of apigenin. We would again hope that most of our patients or clients are consuming these types of foods regularly in their diet, but you can see that there are certain foods that have a higher composition of apigenin, namely in the top left-hand corner of your screen, parsley, dried parsley specifically, having a really significant amount of apigenin per weight. Then you have chamomile has a good bit too, so chamomile tea is probably the one area that some patients are familiar with, that some cancer patients will reach for chamomile tea, maybe partly for a relaxing effect, but also getting sort of the dual benefit here by having these higher levels of apigenin, this polyphenol with these chemopreventive properties. Then fresh parsley, celery seed, celery, oregano, peppermint, so a number of herbs and spices that are containing some good amounts of the compound as well. Okay, so time for academics to application. I'm going to take a quick pause and provide you with a specific reflective exercise or question here to take and convert some of what we discussed today so far in the lesson into applying it into a sort of a clinical scenario. So for this exercise, consider here that while polyphenols exhibit potent anti-carcinogenic properties in vitro, challenges of seeing these effects translate in vivo arise owing to issues with absorption and bioavailability. Based on the content shared thus far and your own knowledge of nutritional science, consider 3 ways in which you would optimize polyphenolic content in a patient's diet, either through food preparation or identification of technologies or delivery systems in dietary supplements. So we'll take a brief pause and allow you some time to work on that specific question and exercise, and we'll come back shortly with the rest of the lesson. Okay, we're back, and next we're going to transition now from this individual nutrients or herbs, many of which were those polyphenols we've discussed, into some specific vitamin sources, some individual nutrients that we'll kind of transition to. And the first 2 you see on your screen fall into these areas that we're discussing, vitamins. And again, those on the line of the course today may be familiar with cholecalciferol, vitamin D3, technically being sort of a hormone or having hormone activities, but for kind of simplification purposes, we'll kind of have it fall under the vitamins, vitamin D3 on the left side of the screen, and then on the right side, delta-tocotrienol, a vitamin E isomer, and that I'll discuss in more detail in the upcoming couple of slides. But we'll begin with vitamin D3. So vitamin D3, clearly an important consideration for patients for a variety of factors around health, of course, calcium metabolism and homeostasis, We know there's a role of vitamin D in immune function, which, again, may have some crossover to its potential for cancer prevention here. A variety of different roles that are important in overall health, but when we're looking at chemo prevention or cancer prevention, certainly an area to look at. We probably think about different sources here. Again, dietary sources, somewhat hard to come by. Certainly, there's some sources in things like salmon, a small amount of D2 in mushrooms. Really want to get to the point where we're looking primarily at vitamin D3, egg yolk. There's a few sources, but of course, primary endogenous development of cholecalciferol D3 comes from sun exposure and conversion through exposure to skin exposure. Number of different pathways beyond that influence that: kidney function and health, liver function and health. So we do see, unfortunately, a relatively epidemic prevalence of vitamin D deficiency, or certainly insufficiency, but certainly deficiency. So I'd, of course, be encouraging everyone with the course today, with our lesson, to be testing 25-hydroxy-D in their patients, looking for ranges minimally of 30, in that pretty wide range of 30 to 100, but I think optimally, the sort of optimal levels that we would normally discuss in integrative health and nutrition would probably fall somewhere closer to 50, 60, or slightly higher. Again, bit of a bell-shaped curve benefit here, but certainly want to have someone better into those ranges of optimal vitamin D levels versus an insufficient state or deficient state. And for our purposes of discussion in the lesson today, really because of the role that vitamin D may play in cancer prevention. So again, another discussion point here in this slide, looking at the title of this particular paper in review, the role of vitamin D in cancer prevention, and really discussed initially some of these same concepts of the prevalence of D deficiency. But really, I wanted to highlight here on the right side of the screen the mechanisms of vitamin D's effects and how they may relate specifically to cancer. So vitamin D and its metabolites reduce the incidence of many types of cancer by, number one, inhibiting tumor angiogenesis, so evidence around that, stimulating mutual adherence of cells, so again, preventing that, the ability for cells to migrate and to break apart and to sort of translocate to other areas of the body, and enhances intracellular communication through gap junctions, so similar idea, keeping cells kind of connected where they should be within local regions, and thereby strengthening the inhibition of proliferation that results from tight physical contact with adjacent cells within a tissue, so that contact inhibition. Vitamin D metabolites help maintain a normal calcium gradient in the colon epithelial crypts, and high serum levels of 25-OHD are associated with markedly decreased proliferation of non-cancerous but high-risk epithelial cells in the colon. So this sort of, that last line or two really relating, of course, more to the potential for vitamin D's influence in gastrointestinal cancer, but you can see really even that the first note or two there: tumor angiogenesis, cell adherence. So all these areas where are important to prevent that initial development of a cancer cell and then its prospective migration to other areas or metastases. So really important to kind of consider, all these areas, the active form of vitamin D, 25-OHD, converting over to 125-OHD, again, which I mentioned before, has some influence around liver function, so something to consider supporting in patients. It's more than simply just saying, encouraging patients to get out in the sun, because then there's, again, that potential for how much appropriate sun exposure is relevant for a patient that, we're looking at chemo prevention and not inducing any type of damage from excessive amounts of sun or radiation exposure through sun exposure and the possibility around sort of a melanoma or skin cancer development. So enough vitamin D to kind of play its role in a couple of these different mechanisms we just discussed. And we can look a little further at vitamin D because it has a series of additional benefits. Namely, the additional benefits are really, we've somewhat hit on those a little bit so far, but the ability of vitamin D to induce terminal differentiation apoptosis. So again, to go back and to have that specific benefit of inducing apoptosis in the cell that again may be damaged already. So let's transition from vitamin D over to tocotrienol. So delta tocotrienols again may be familiar to some, maybe not to others. Basically talking about a form, an isomer of vitamin E. And so everyone's probably familiar traditionally with vitamin E as sort of a family of vitamin isomers. And historically kind of the focus being on the tocopherols and on primarily alpha tocopherol from some early evidence and research. But really as time has gone on, there's been more of an emphasis and more potential focus around tocotrienols and their again potential of chemoprevention. So the tocotrienols are a vitamin E fraction, and you have all the same alpha, beta, gamma, delta isomers that you have with the tocopherols, and so the tocotrienols themselves appear to have possess some additional benefits above and beyond the tocopherols, and in this case specifically having some pretty interesting and promising anti-cancer properties and effects. So we'll look here at the Journal of Cellular Physiology around the anti-cancer properties of tocotrienols, mechanisms and molecular targets. And some of this may be owing to the actual structure of tocotrienols. They do provide a number of different benefits that are slightly different than alpha tocopherols. So, typically talk in physiology of structure dictates function, and tocotrienols have a sort of slightly different structure than the tocopherol families, and has a shorter tail kind of structurally than tocopherols, which allows it to move a bit more rapidly for imparting some of its effects, namely some of its antioxidant effects, and really its antioxidant anti-inflammatory effects around lipids, so inhibiting lipid peroxidation. And important because we're looking at really kind of cell membrane phospholipid protection here. So that's some of the influence that the tocotrienols have. Here, when we're looking at the specific study and the comments from the researchers, the highlighted section discusses that several in vitro and in vivo studies pointed out that tocotrienols also exert a significant anti-tumor activity in a wide range of cancer cells. Specifically, tocotrienols were shown to exert anti-proliferative and pro-apoptotic effects, and to reduce the metastatic or angiogenic properties of different cancer cells. Moreover, these compounds were reported to specifically target the subpopulation of cancer stem cells known to be deeply involved in the development of resistance to standard therapies. So a lot to kind of unpack there, but essentially some of the same types of pathways that we've talked about that are involved in the hallmarks of cancer. So proliferation or unregulated proliferation, tocotrienols seem to have an anti-proliferative effect. Pro-apoptotic, we've seen again the evading apoptosis is a hallmark of cancer development. Tocotrienols appear to have a pro-apoptotic effect, basically inducing that programmed cell death. Reduction in metastatic properties, so again, metastasis, those cells breaking off into, again, adjacent areas or further areas away of the local tissue, tumor tissue. Tocotrienols seem to address the metastatic properties and also angiogenic properties, so that development of new blood vasculature to provide nutrients, oxygen to cancer cells. Again, tocotrienols appear to have some activity against angiogenesis as well. But really, kind of impressingly and interestingly here, they've been reported to specifically target the subpopulation of cancer stem cells. So cancer stem cells are an area of expressed interest in the research now. This paper is from August of 2018. So this is recent research showing that these specific vitamin E isomers of tocotrienols have this effect to target the cancer stem cells. Really important because I think there's certainly established and growing evidence that it's cancer stem cells that may sort of stick around even when a primary tumor has been dealt with either by surgical intervention radiotherapy, chemotherapy, and that's what is in part the underlying challenge or issues of resistance to standard therapies, when it seems like these therapies may be working, but you've got a cancer that either recurs or is difficult to actually completely eradicate. So maybe an opportunity here, as we've mentioned in prior slides in this lesson, for tocotrienols to impart that benefit synergistically, to work maybe with some of the standard therapies to make them more effective as well. The bottom highlight on this page shows and speaks to what the researchers highlight as promising results that, together with the safety of tocotrienol administration in healthy subjects, suggest that these compounds might represent a new chemopreventive or anti-cancer treatment, i.e. in combination with standard therapies strategy. So a new anti-cancer treatment strategy or a new chemopreventive strategy. So really promising results. A review article by the researchers just published in last year. Really interesting, and again, probably an area that I would assume not a lot of clinicians are familiar with in terms of the vitamin E isomers. So one area to begin to explore more, certainly more research is needed. I know there's some research being conducted in Denmark around some ovarian cancer trials using tocotrienols, various dosing ranges as well, but in a supplement form. Again, tocotrienols are primarily from a food basis, found in rice and palm. Very sort of relatively small amounts in those two food sources, and there are as much or maybe more in those two food sources tocopherols. And there does seem to be some evidence and that the tocopherols themselves block some of the protective or positive effects of tocotrienol. So there's somewhat of an antagonistic sort of blocking of these positive effects. So that's the interesting part, and as some of this evidence is shown when we're extracting here, we're looking at things like, and you can see it in the left-hand side of your screen, one other source is annatto seeds from the annatto plant. So you may be familiar with annatto as a food coloring. It's used to sort of color mustard, sort of has that sort of orangish, that type of hue, orangish yellow, but that's a sort of a pigmentation use in food processing. But the annatto seeds themselves are a highly rich source of tocotrienols, primarily gamma and delta tocotrienols, and are actually void of the tocopherols. So they are able to sort of circumvent that potential for tocopherols to interfere with the positive and anti-inflammatory, antioxidant, anti-cancer properties of tocotrienols. So, I mean, looking at either from a dietary perspective, may be a bit challenging to get annatto in if we're talking about chemoprevention, or using this in concert with an anti-cancer treatment, a standard therapy, or we're probably looking at dietary supplementation of the tocotrienols. Okay, next on to zinc and copper. So relationship here between two specific nutrients, two minerals, and important because there is some evidence around not only the individual status of these two in health, but also specifically to their ratio in the prospect to be used as a clinical marker. So the copper zinc ratio as a clinical inflammatory nutritional biomarker, it can actually and been shown to be a significant predictor of all-cause mortality in elderly population over 70 when you have a high copper to zinc ratio. So several studies have also shown that plasma copper concentrations are increased in various carcinomas. Zinc acts as a cellular growth protector, including growth of neoplastic cells. And zinc deficiency has been demonstrated to be involved in several stages of malignant transformation. So it's a relative ratio too. So high amounts of copper being associated with, again, various different types of carcinomas, and low status of zinc. Of course, of those two, again, having that relationship, copper high, zinc low, having really, again, more of a potential for opening the door for cancer development. So RDA for copper is 900 micrograms a day for adult men and women. The RD for zinc in the U.S. Per the Food Nutrition Board Institute of Medicine is 11 milligrams per day for adult males, 8 milligrams per day for adult females. And most patients can get the sources of these two minerals in their diet. Some of the challenges that the foods that are high in each of these nutrients are the same foods, that you actually have, again, some animal-based proteins and meats that have are good sources of zinc and copper. Zinc probably is primarily, associated with oysters as one version, oysters and other crustaceans or seafood. But really, again, looking to balance out the ratio. You can certainly do lab markers and biomarkers to test for serum levels of zinc and copper, or ceruloplasmin as this sort of storage form of copper as well, and to kind of see where these ratios fall. So how do we balance zinc and copper to ensure that we have adequate zinc status and adequate or sufficient but not excessive copper status as well? And really that's just by looking through, looking at a patient's diet, considering what kind of sources they are. And you can see in this next slide here, we've got on the left-hand side of the screen, dietary sources of zinc. On the right-hand side of the screen, dietary source of copper. And of course, as I mentioned, one of the top sources of both are oysters. So not that oysters wouldn't be a bad food to incorporate, but they're going to contain both zinc and copper. But you can see on the left-hand side, some additional sources of zinc include beef, crab, lobster, pork chop, so a number of different types of meat and seafood, baked beans, chicken, yogurt, cashews. On the right side, some crossover here, but a lot of the dietary source of copper will come from, in addition to the oysters we mentioned, whole grains, beans, nuts, a number of organ meats contain pretty high sources of copper as well. So just about, kind of balancing out the dietary intake. And then if we're looking at sort of the biomarkers to look at the ratio, a balanced copper to zinc ratio is between 0.7 to 1.0 based on several studies. And again, just to kind of reiterate where that ratio kind of comes into play here, is that a balanced ratio helps support and maintain the immune system in an appropriate manner, helps resisting infectious diseases, of course, zinc being associated with sort of immune system generally, has the potential to be used as a clinical indicator of oxidative stress. So back to that last slide where oxidative stress, clinical nutritional biomarker for inflammatory status. So we'd like to be able to see that ratio, 0.7 to 1.0, copper zinc ratio. So we want to kind of bring it all back in together and to be able to essentially tie things back of what we've discussed not only today in Lesson 3, but in both Lessons 1 and 2 as well, around how the variety of different dietary patterns, general recommendations of dietary intake, and specific individual nutrients, herbs, botanicals, all kind of play a role in how they specifically can all target and affect the variety of hallmarks of cancer. So on the very last slide here, we see the hallmarks of cancer, and now how each of these different considerations around nutrition will be able to influence these hallmarks. So as we work our way through, important to be able to see a number of these here. So we can go through a series on the screen here, but for example, two of the things that we talked about early on, the increases in dietary fiber, increases in fruit and vegetable intake, are both going to help support and kind of influence tumor-promoting inflammation. They generally have an anti-inflammatory effect in the body. And again, not all these are exclusive to one specific hallmark. Several of these will have some crossover, but certainly having adequate amounts of fruit and vegetable intake and dietary fiber intake are going to be important for reducing tumor-promoting inflammation. So those were the two dietary habits or general approaches or recommendations. Now, what were the things we mentioned to kind of decrease? And those were processed meats and sugary drinks. And both of those, again, can have various effects. Sugary drinks especially, though, kind of influence the hallmarks around deregulating cellular energetics. So that's another area where the modulation of diet can come into play. We also talked about decreasing alcohol intake in Lesson 1. And alcohol can have some damaging effects. Again, can be multiple different hallmarks here. Certainly could be around cellular energetics, but also could be around so the damage to genome stability, so genomic instability and mutation by long term. Some of the metabolites of alcohol can be damaging as well. Then we kind of shifted into dietary patterns. What are some of the dietary patterns associated with chemoprevention, and which of the hallmarks of cancer that we see here would they influence? So we can go back to a few. For example, the Mediterranean diet, again, may affect multiple pathways on the hallmarks of cancer here, but because of its effects on regulation of blood sugar, it would be important to influence the deregulating cellular energetics effect. Of course, it can also support the immune system. It may decrease that tumor-promoting inflammation by higher omega-3 fatty acid intake, by increases in those anti-inflammatory fruits and vegetables that we mentioned as well. So a number of things that could be influential there. From Mediterranean diet, we looked at some things like ketogenic diet, and the ketogenic diet really specifically is influencing cellular bioenergetics. So the hallmark of cancer being those deregulated cellular energetics. So because of that decrease in that glycolytic flux, increasing that fatty acid and ketone metabolism, not only for cell energetics, for ATP production, but also because of some of the anti-inflammatory and cell signaling properties of ketone bodies. Those things would all be influential here. Again, a number of different hallmarks of cancer. We also talked about caloric restriction. So caloric restriction, same thing, can influence cellular energetics certainly, may also influence genome instability and mutation. But another area of caloric restriction may be its ability to influence the hallmark of cancer, the enabling of replicative immortality. So again, multiple areas here, but caloric restriction can certainly be influential there. We talked about broader caloric restriction, intermittent fasting, various fasting practices that can help with some other areas that, again, aren't even necessarily highlighted here, but caloric restriction in some degree of whether it's prolonged or intermittent would certainly influence some things like autophagy that we saw in previous slides and previous lessons as well. The other piece here after caloric restriction is that we shifted then from dietary patterns broadly into individual constituents, individual nutrients, herbs that can influence these hallmarks of cancer. So we discussed originally curcumin, and again, it has a very potent anti-inflammatory effect, so would influence the hallmark of tumor-promoting inflammation in cancer. We talked about EGCG, and it has, again, a variety of benefits that are around this entire wheel of the hallmark of cancer, but one of the big ones would be its ability to help address that induction of angiogenesis, to again, prevent or inhibit some of that angiogenic activity. So again, there's multiple other pathways here. After EGCG, we also discussed sulforaphane, and sulforaphane also, again, decreasing tumor-promoting inflammation, influencing the activation of invasion and metastases. So we saw a few slides earlier in today's lesson that it influences metastatic activity as well. Also has some benefit around that genomic stability and mutation property. So a number of things here. In addition to sulforaphane, curcumin, EGCG, we discussed quercetin, and quercetin had some immunomodulatory effects, so it would influence the hallmark of avoiding immune destruction, but again, had some anti-inflammatory effects, had some ability to affect angiogenesis, metastasis, cell stability, so multiple areas here. After the quercetin, we also discussed apigenin. And apigenin, again, we mentioned multiple pathways here, but and for our purposes, we'll talk about resisting cell death. When the influences on the hallmarks of cancer, apigenin has the ability to kind of induce apoptosis, prevent that evasion of appropriate cell death that cancer cells often exhibit. And after apigenin, we also discussed today the couple of specific vitamins or nutrients, vitamin D3 and tocotrienols, or T3, sometimes see that referred to, not to be confused with thyroid hormone, but D3 again has an immunomodulatory effect, so we could say that it will influence the hallmark of cancer around avoiding immune destruction. But again, multiple pathways by which D3 seems to have an effect. And tocotrienols again, can have multiple pathways that they're influencing, sustaining proliferative signaling. Tocotrienols appear to have some activity there, evading growth suppressors, so influencing tumor suppressor activity in tocotrienols. They're certainly having an anti-inflammatory effect, so tumor-promoting inflammation being relevant there as well. And then finally, we talked about the zinc to copper ratio, and again, its influence on multiple hallmarks of cancer. Talked about it being really kind of regulatory for certain things, inflammatory effects, that replication or proliferation effect, but evading growth suppressors, so also having some activity there. So many of these compounds, dietary patterns, general recommendations of food intake that we've discussed in lessons 1, 2, and 3 will affect multiple hallmarks of cancer, and that's the important part. So perhaps the takeaway is that we want to be able to employ not just one of these, but a variety of different therapeutic effects, which allow for some variety in the patient's nutritional or dietary intake for chemoprevention. Do any one of these work better than another? Not necessarily, so it may be a matter of preference and different pathways we'd want to address, but certainly having some of these dietary constituents and some basis of the dietary pattern will be ideal. And again, there's always some crossover here. So if you're eating a diet that's high in fruits and vegetables, presumably we have high dietary fiber. If those fruits and vegetables consist of things like cruciferous vegetables, broccoli sprouts, that's going to have high levels of sulforaphane. It also assumes that maybe it has high levels of some herbs like parsley, so we'll get the apigenin in. Potentially you could have some high amounts of onions, apples, other foods that have quercetin, so we'll have that compound as well. If we're eating a variety of different, through, like, for example, the Mediterranean diet, again, touches on some of them using herbs and spices liberally, so getting turmeric in, being able to have an appropriate zinc to copper ratio with those dietary intakes, and of course, something like the Mediterranean diet or keto, avoiding a lot of processed meats and sugary drinks. Alcohol may be decreased, certainly in keto. Mediterranean diet might see just a general bit of dietary intake around typically red wine, but again, having some balance there is important, and being able to, when appropriate and necessary, supplement with certain things like higher intake of things like D3, potentially the tocotrienols as well. So this really concludes Lesson 3 and the entire Nutrition for Cancer Prevention course. I want to thank everyone for taking this course, and certainly want to be able to hopefully helped address some of your questions or thoughts and provided you with a framework by which you can work with your patients around chemoprevention through diet. So thank you for joining me. This is Jason Bosley Smith for MUIH. Bye, everybody.